Okay, so DNA is often used as a metaphor in common speech to reference things that are basic or very internal, something that builds on something else. That's exactly what it does in biology. So it is the blueprint for which we make our proteins and for which we function as things that are alive. Everything that is alive has DNA. Now, how we discovered what it was is actually a very interesting story. So we'll go through some of the discoveries of DNA and what we learned with each discovery. So we're going to start with Frederick Griffith. So in 1928, what he did was he looked at pneumonia, two different strains, one of which was which would cause death, and one of them which wouldn't. Okay, and so in the first experiment, or as controls, he used this strain of pneumonia which was lethal. It caused mice to die, and he used this other strain which did not cause mice to die. They could recover from the infection. And what he did is he killed the virulent strain that killed mice, and after he killed it and then put it in there, the mice lived. But when he mixed a heat-killed virulent strain with a non-virulent strain, the mice that were injected with this mixture died. So the conclusion was that, something is going on here. Something is being transferred or picked up from the dead strain and moved into the non-deadly strain. Okay? So another experiment by Avery, MacLeod, and McCarty in 1944 repeated that experiment using purified cell extracts. So they took the cells and purified them into different parts. So one thing they did was they destroyed the proteins. Okay, by destroying the proteins and then going through and leaving everything else the same, the experiments had the same results. But when they destroyed the DNA, the mice lived. So from this, they concluded that, there must be some transfer then of the DNA from the dead one to the live one, which causes the non-virulent strain to become virulent, so the non-deadly strain to become deadly. So this was the first clue that DNA is very important. It has some sort of instructions or some sort of changing ability within it. Alright, so the next experiment, Hershey and Chase in 1952. What they did is they invested bacteriophages. And bacteriophages are viruses that infect bacteria. Okay, so here are the viruses. That's what they look like. This is the bacteria. They infect them. Okay? And what he did, so he labeled protein and DNA of a bacteriophage with some radioactive material. And then he allowed them to infect the material, the bacteria. In one experiment, he used sulfur, which would attach to the protein part of the virus. And when he looked at the bacteria that were infected, that protein was not within it. Okay? When he did the same thing with phosphorus, which does get uptaken by DNA, they looked at the bacteria, and yes, they had that sulfur bacteria in it. So it must be the transfer of DNA was the conclusion from this. It must be the transfer of DNA which caused, the infection by the virus. All right, so DNA is a nucleic acid, and we have established that before. It has nucleotides, which are made of a 5-carbon sugar called deoxyribose, a phosphate group attached to the 5' carbon of the sugar, and a nitrogenous base. Now, 5', that may sound funny, but on this 5-carbon sugar, each of these carbons has a number associated with them. This is the 1', 2', 3', 4', and 5' carbon. This will be important later. On the 5' end, you have a phosphate group. Okay, so that's all that means. Then you have a nitrogenous base. And there are 5 different types. 4 of them are found in DNA: adenine, guanine, cytosine, and thymine. And then they have a free hydroxyl group on the 3' end of the sugar. So this is the 3', there's the OH there. Now between nucleotides you have what's called a phosphodiester bond. So that's where this phosphorus molecule will bind to the 5' carbon on one end and the 3' carbon on the next. So the chain of nucleotides has a 5' to 3' direction. Basically, the 5' is on top, the 3' is on bottom. And that's where the next phosphodiester bond would be. Now, Erwin Chargaff did some experiments where he purified DNA, and he found that the amount of adenine was the same as the amount of thymine, and the amount of guanine was the same as the amount of cytosine. Now, these nitrogenous bases are called purines, and these are called pyrimidines. So you had an equal amount of purines as you had pyrimidines. Okay, and they were always in equal proportion. We'll talk about why in a minute. Rosalind Franklin also helped to determine the shape of DNA by looking at X-ray diffraction studies. So this is what an X-ray diffraction looks like. Based on that, I don't see it, but based on that, they can determine that it was helical. Okay? And using Maurice Wilkins' DNA fibers, they discovered that the molecule has a diameter of 2 nanometers and makes a complete turn of the helix every 3.4 nanometers. Okay, so it's really, really small. But it turns, okay? So it has a helix. It's like a ladder that's twisted. Now, Watson and Crick, James Watson and Francis Crick. These are 2 guys that put everything together. They didn't actually do any experiments, but looking at Rosalind Franklin's, Chargaff's rules, the structure of DNA, and all these other previous experiments, they proposed the double helix structure in which these nitrogenous bases bound to each other. And so they are credited with the discovery of the structure of DNA. So this double helix is 2 strands of polymers of nucleotides. The phosphodiester bond is the backbone. And then they wrap around one axis, and they're anti-parallel. That means one goes in one direction, and then on the other side you have the other strand going in the opposite direction. What that allows it to do is for the nitrogenous bases to make hydrogen bonds with each other. Okay, if they were both in the same direction, then they wouldn't be able to make these bonds. So, guanine and cytosine are able to make 3 hydrogen bonds. Adenine and thymine are able to make 2 hydrogen bonds. And this is like the rungs on the ladder then. All right, so the next question was, well, how is DNA replicated? How is it when you make 2 strands out of 1 strand? And these were the different options. You could have a conservative model where the old strand was kept intact with 2 old strands, and the new strand was made with 2 new strands. Or you could have a semi-conservative model where you had a new strand added to one of the old strands and a new strand added to the other old strand. So the new strand was a combination of new and old. Dispersive is another model where both strands had pieces of old and new. Now, based on experiments, they discovered that the semi-conservative model is the correct way that it replicates. So DNA replication then requires 3 things. First, it requires something to copy. So that's going to be our parental DNA molecule. That's going to be our blueprint for what to do. It requires something to do the copying, which is a bunch of enzymes. Okay, these enzymes help break things apart, help add things on it, makes it go very quickly. And then you need building blocks to make the copy. So you need nucleic acids to add on to there. So we break DNA replication into 3 different processes. The first is called initiation, and this is when you unwind DNA and start, right? The second is elongation, where you're adding nucleotides on top of each other to make a new strand. And then the third is termination. Once it's all completed, you separate the 2 new strands from each other. So when replication occurs, based on the enzymes, and the orientation, they can only add on to one end, and that is on the 3' end. So again, remember we had 1, 2, 3, 4, and 1, 2, 3, 4, 5. Okay, this is the 5th carbon right here. And you can't add one onto the 5' carbon. So you can't add a new strand on here. You have to add it onto the 3' end. Okay? And so bases are synthesized in the 5 to 3' direction. So you put the 5 on top and the 3 on bottom. So that means on this one, which is going the opposite direction, you have to add it on this side, on the 3' end. And this requires an enzyme called DNA polymerase. Okay, and DNA polymerase adds the nucleotides to the new strand. So DNA, the way it does this, it matches existing DNA bases with complementary nucleotides. So if you have an adenine here, it'll add a thymine next to it. And it adds the new bases to 3' end, again in a 5' to 3' direction. So 5' over here, it's adding it onto the 3' end. And it requires, to get started, a primer of RNA, and that is done by a different enzyme called primase. So we have prokaryotes and we have eukaryotes, and their replication processes are slightly different. So we're going to start with eukaryotes. The E. coli model is a circular singular chromosome, if you remember that. Replication begins at the origin of replication and then goes in both directions around the chromosome until you have 2 complete DNA molecules, chromosomes. It ends at the termination site. Now, E. coli has 3 DNA polymerases, so 3 different types that will add on nucleotides. DNA polymerase 1 acts on the lagging strand. We'll talk about what that is in a minute. And it removes primers and replaces them with DNA. DNA polymerase 2 is involved in DNA repair processes, and DNA polymerase 3 is the main replication enzyme. All of them have 3' to 5' exonuclease activity for proofreading. And DNA polymerase 1 can also do it in the opposite direction, 5' to 3'. Alright, so there are a few other enzymes very important other than DNA polymerase in this process. Helicases unwind the DNA. Okay, so they open it up so that these DNA polymerases can go in there and add new nucleotides. Single-stranded binding proteins will then bind to the unwound DNA to keep it from winding back up. And then topoisomerase prevents supercoiling. So if you continue to unwind this, it's like if you're taking 2 strings and trying to unwind them from a larger piece of string, it will cause supercoiling. Basically, on the other end, it'll start to coil. And so topoisomerase then prevents that supercoiling by cutting and rebinding it. As I mentioned before, it's semidiscontinuous. Or no, we meant it was something else. All right, so semidiscontinuous means that it can't go all in one smooth motion. So because it can only go in one the one strand, which is called the leading strand, that's very easy for it to just continue adding as you unwind the DNA. But the other strand, it's going away from the opening, so it will go the opposite direction, and then it'll have to keep coming back every time that it's unwound more. So this is called the lagging strand. It does not happen as quickly as the leading strand because it has to keep putting on more and more primers and as the DNA unwinds. Now these fragments then are called Okazaki fragments, named after the person who discovered them. So the point at which you have this opening and where you're starting to add nucleotides is called the replication fork. DNA primase is what can then add a primer on. RNA polymerase makes the RNA primer. Okay. How many primers will you need for the leading and lagging strands? Well, the leading strand you only need one primer because then it can go continuously in one direction. But the lagging strand you will need multiple primers, okay, from primase. Ligase then, after DNA polymerase comes and removes the RNA primer, there's still a section then where it still needs to be bound, and DNA ligase will go and bound that little section and seal the nicks. Gyrase, DNA gyrase then unlinks the copies when they're all finished. So once you complete 2 strands, you need gyrase to go in there to unlink them. Okay, there it is. Here it is on with both showing both at the same time. Leading strand just adds DNA polymerase, just adds nucleotides. The lagging strand though needs these primers, and after the polymerase then removes the primers, ligase will seal the nicks. Alright, eukaryotic replication is a little more complicated because it has much more DNA and it has a linear structure. So enzymes are the same except for telomerase, which we're going to talk about later. It also requires new enzyme activity for dealing with just the ends, so that's what telomerase is for. So some of the differences include multiple replicons, or multiple areas where the replication has to begin. Okay, whereas in prokaryotes you can just start at one place and go all the way around. Eukaryotes, you start in multiple places and then they all kind of meet when they're done. The initiation phase of replication is also more complicated. It requires more factors to get started. And this primase includes both DNA and RNA polymerase. Alright, so now we're going to talk about, so we did replication, we're going to talk about something called the Central Dogma. And this is basically it. This is how we make proteins, and proteins are the functional parts, for the most part, in our cells, and thus in our bodies. So to get proteins, you have to start with DNA. With DNA, you make RNA, and with RNA, you're able to make protein. The process of making DNA to RNA is called transcription, and the process of making RNA to protein is called translation. Okay? So in transcription, this is like you're transcribing something. Really, you're just kind of making a copy. And that's, DNA and RNA are so similar, it's like you're making just a copy, a small piece of a bigger manuscript. You're just going to make a little transcription of it. Translation, though, proteins are made of amino acids, which is a much different structure than nucleotides. So here you're translating RNA, looking at it, and this would be like translating into a new language and making a different functional product. So in DNA, we have our 4 bases: adenine, guanine, cytosine, and thymine. Adenine pairs with thymine and guanine with cytosine. In RNA, you have the same thing, except instead of thymine, you have uracil. Otherwise, the pairings are the same. adenine with uracil, and guanine with cytosine. In protein, you have 20 different amino acids, which we have already talked about, and each of them have different molecular properties. The codons of mRNA, and codons are 3 nucleotides. So an adenine, a guanine, and a uracil would make a codon, and these code for different amino acids. Okay, so you need a few different things for each of these processes. First, you need a blueprint or a template. Then you need the machinery, something to carry it out. And finally, you need building blocks. Okay, so DNA, in transcription, DNA is the blueprint. The enzyme is RNA polymerase, which adds the nucleotides. And the nucleotides are the building blocks. The process involves initiation, elongation, and termination. In translation, the messenger RNA is the blueprint. The ribosome is the enzyme, and the amino acids are the building blocks which you're going to put together. And those are carried on transfer RNA. And the processes for that are also initiation, elongation, and termination. So, we've talked about RNA before. There are 3 different types. There's actually more types than this, but these are the 3 main types: messenger RNA, ribosomal RNA, and transfer RNA. And each of them is important for transcription and translation. All of them are also synthesized by the DNA as well. So by reading the DNA, you can make each of these. Now, Francis Crick and Sydney Brenner determined how the order of nucleotides coded for the amino acids. And like I said before, it's a three DNA nucleotides each hold 3 different bases, and those 3 bases are read to code for an amino acid. And what they did to learn which was each is they inserted or deleted certain nucleotides and figured out what the amino acids made. And this makes what's called a frame shift mutation. We'll talk about that later too. Alright, so from this genetic code, they were able to determine different types of codons, what they meant. So there were a few different stop codons. So a uracil, and a adenine was one of them, and these are the other ones, UGA and UAG. There was also a start codon, AUG, and there then were many different other types of amino acid codons for the other ones. The code is degenerate, which means that some amino acids are specified by more than one codon. And it is practically universal, meaning almost all living organisms have the same amino acids. And here they are. And here is the different codes for each of them. So leucine has 4 different combinations of RNA. And so this would be a cytosine, uracil, and uracil, and that's going to make a leucine. But anyway, we'll go over some more stuff like this in class. So prokaryotic transcription, the initiation does not require a primer, but it does require a promoter, and then a start site and a termination site. So at the promoter, you form a recognition of the binding site for the RNA polymerase. And this is found upstream of the start site. So the RNA polymerase first finds the promoter. It doesn't transcribe it, but then it finds the downstream of that, it finds where to start. And this is also asymmetrical, which indicates the side of initiation and the direction. So it's going to be different for going one way or the other. Elongation then, once it's past that, it will unwind a little bit of the DNA and grow in the 5' to 3' direction as these nucleotides are added to each end. When it unwinds, it forms this little transcription bubble. Okay, and so it starts adding these RNA nucleotides on the end, and then it kind of recloses after it adds those RNA nucleotides. After it is passed, then the now transcribed DNA is rewound and it leaves the bubble, as I just said. All right, finally, termination occurs, and that's marked by a stop sequence. So phosphodiester bonds no longer are formed between the nucleotides. And then the RNA-DNA hybrid, where they're bound together, starts to dissociate. The RNA polymerase then releases the DNA, and the DNA rewinds. Now, some of these RNA pieces then, it would be one long piece, but some of these RNA bases will bind to each other, just kind of like the double helix of the DNA, but much smaller. And then they will make these structures called hairpins, and the hairpins help them to release. All right, prokaryotic translation then, it is coupled with transcription. So it kind of occurs before transcription finished, translation has already started, and you start getting amino acids moving on to them. The operon then is the group of functionally related genes, and these have multiple enzymes that can be regulated together. So eukaryotic transcription is a little bit different. RNA polymerase is used. RNA polymerase 1 is used to transcribe RNA. RNA polymerase 2 transcribes mRNA, and RNA polymerase 3 transcribes tRNA, and also some other smaller RNAs. And each RNA polymerase recognizes its own promoter to be able to make each of those functional products. So transcription starts in eukaryotes as a series of transcription factors. These are necessary to get RNA polymerase 2 enzyme to a promoter and then to initiate gene expression. Interacts with RNA polymerase to form an initiation complex. Okay, together, so you have these promoter transcription factors. They interact with RNA polymerase 2 to start the initiation complex. Termination sites aren't as well defined, but there's basically a sequence which causes all this to dissociate. Once mRNA is made, then there's some modifications that need to be done to it in order for it to be coded into a protein. So first, a cap and a tail is put on it. On the 5' end, they put on a cap. On the 3' end, you have a poly A tail, so lots of adenines. And some sequences in the middle there are taken out. The ones that don't code for anything are called introns. And this is done by an enzyme called the spliceosome. So introns are the non-coding sequence, and the ones that are left are called exons. So there are two different things which are interacting. The small ribonucleoprotein particles, or SNRPs, they recognize the boundaries between the introns and the exons, and then they cluster with other proteins to form the spliceosome, and then they remove the introns. Now, what allows for us to have less? So it would be that every gene would need to have its own mRNA, which would produce a unique protein. But we can make different proteins out of the same sequence by shifting around the different exons. So if this is the order in which they are transcribed, 1, 2, 3, 4, you can have alternates, and this is called alternative splicing. So you could have 1, 2, 3, 1, 2, 4, you could have 4, 2, 1. And basically, you can mix those around to make different proteins. And that's why we make 80,000 different mRNAs in the human genome, but only 25,000 genes are needed to do that. Finally, you have your transfer RNA, and the transfer RNA brings the amino acids to the ribosome so that they can add amino acids together. The aminoacyl-tRNA synthetase adds amino acids to the acceptor stem of the tRNA. And then an anti-codon loop, which reads the mRNA, then it corresponds with specific amino acids. Each aminoacyl-tRNA synthetase recognizes only one amino acid, but several tRNAs. So that's again because they have. We showed that before on the graph of the amino acids. A charged tRNA has an amino acid using energy from ATP, and it can undergo a peptide bond formation between amino acids. So it doesn't need any additional energy. It has everything it needs on the charged tRNA. Ribosomes don't verify the amino acid attachment to the tRNA. So if there's a mistake there, then it's not going to be corrected by the ribosome. The ribosome has multiple tRNA binding sites, the P site, A site, and E site. The P site and the A site is where the amino acid is being transferred, so it comes in the A site, and then the amino acids on the P site then transfer onto the A site, then everything shifts, or the tRNA that's no longer in use exits out the E site, and then a new one comes in on the A site. The ribosome has two primary functions, one to decode the mRNA, and the second is to form the peptide bonds between the amino acids on the tRNAs. And peptidyl transferase is the enzymatic component of the ribosome. So this is the part of the ribosome that does the enzyme that forms the bonds between the amino acids. Alright, so translation then in prokaryotes starts at the initiation complex and includes an initiator tRNA charged with an N-formal methionine. Okay? So an N-formal methionine is the first one of every amino acid. It goes on there on the small ribosomal subunit. And then a ribosome binding sequence of mRNA positions it correctly, and the large subunit then is added on top. And so it's now bound to the P site, ready for another one to bind to the A site, and that's what happens next. So initiation in eukaryotes is similar, except the amino acid is not that really long one, it's just methionine, and it has a more complicated initiation complex, and it doesn't have an RBS, which is the small subunit that binds to the 5' cap of the mRNA. Elongation then occurs as amino acids are added. The peptide bond then forms between the amino acids, and then another one can be added as a cycle. So if we start here, we have this double chain with a GTP, and which can be used for energy. It then moves. The elongation factor is lost, and then the amino acid is transferred from, the amino acid chain is transferred from the P site to the A site, and then it moves down, and the next one, the one that no longer has the amino acid chain, goes to the E site, and then it exits out, and then another one can go in there. Alright, in translation there are fewer tRNAs than codons, and this is what we call wobble pairing. So it allows a less stringent pair between that third one. So it doesn't matter if it's a G, it doesn't matter if there's a C or U or a G here. It doesn't have to be perfect, and so that's why you can have different codons, anticodons, coding for this same amino acid. And so you only need 20 tRNAs instead of 60 for the same amino acids. And again, that's why we have repetition here in the different types of codons which code for the same amino acid. Alright, translation terminates as elongation continues until it gets to a stop codon. A stop codon basically makes everything dissociate and everything releases. Alright, so mutations can occur where you have a single base which can cause a lot of things to change, or it can cause nothing to change. So a base substitution is where one base gets switched out for another. Okay, if it's one of those that codes for the same thing, so you have a C going into this T spot, that's going to code for a G on the other end. It's still going to make proline because of that wobble pairing. So this is called a silent mutation. But sometimes you can have a missense mutation where it changes the amino acid, and that's what happens here. Okay. So if you change this one right here, it was originally a GC to an AT, that can cause this to become a stop codon, and now you have a shorter amino acid and probably not a functional protein. Frameshift mutations are adding or deleting of a single base, so you don't have a substitution. Instead, so you have ATTCAC. Here, we would add another DNA base. Okay, if you add another DNA base there, then it could shift the whole thing, and now the whole thing is going to code for different amino acids, and that's going to be very devastating to the function of that protein. Okay, and some of these cause disease, such as Huntington disease. You also have chromosomal mutations, which can change the structure of a chromosome. If part of the chromosome is deleted or lost, or you can have part of the chromosome duplicated and copied, you can have part of the chromosome flipped around. And then you can also have part of the chromosome move to another area. Okay, and each of these can have consequences. So one difference between chimpanzees and humans is an inversion of one of the chromosomes. Other than that, most of the chromosomes are similar. Now, mutations are the starting point for evolution. A mutation can provide a novel characteristic or novel protein. Too much change or too much mutation, however, is harmful, and that person will die and be selected against. So balance must exist between the amount of new variation and the health of the species. All right, so I said we're going to talk about telomerase. So one of the things is telomeres, which are the ends of the chromosomes. These are basically like caps to prevent deterioration and maintain the linear integrity of the chromosome. So over time, after every time you replicate, they get smaller and smaller because the last section of the lagging strand can't be replicated. So that last little bit can't be replaced because it has a primer on there. Okay, and so it gets shortened. So what telomeres do is they compose short repeated sequences to extend that last little piece so that you can preserve that last little end. There are other things, however, that can cause problems to your DNA. Some of them may be due to replication. So DNA polymerase has a proofreading ability. It is able to see if it makes a mistake sometimes. But mutagens will cause, these are radiation and chemicals, which will cause an increase in the number of mutations above what is normal. And if you have too many mutations that can't be repaired, then those can cause problems. So DNA repair is very important, and it's there's evidence of that because DNA has many different repairing mechanisms. Alright, most DNA repair falls into two categories. You can have specific repair, where it's a specific kind of lesion in the DNA, or you can have non-specific, where it can just correct whatever's messed up. It doesn't matter, it'll just take it out and redo it. So photo repair is an example of a specific repair mechanism. And so this happens if you're exposed to UV radiation from light. It'll cause two thymines that are together to stick to each other rather than to bind to the adenines on the opposite side. Okay, so a photolyase will go and actually break that bond and recorrect them. And so this is very specific to thymine dimers, which is why it's a specific repair mechanism. Excision repair, they will actually, it doesn't matter what's going on as far as this damaged or incorrect base or something going on here. It will just come, remove it, and then get the DNA polymerase to go in here and repair it, add new ones. So first you have to recognize, then remove, then resynthesize that information on the undamaged using the undamaged